Hi everyone, my name is Rodrigo Isaac Rojas Jimenez. I am from UPIBI, from the National Polytechnic Institute, and in this occasion I will be talking about these nanoparticles, these silicon dioxide nanoparticles, mesoporous nanoparticles that we not only synthesize but also characterize many of their properties. As an introduction, I would like to remember famous physicist Richard Feynman, who stated there's plenty of room at the bottom, obviously referring to the quantum revolution. And this sparked the newly, in 1959, the newly nanoscience and nanomaterials area. Today, nanomaterials are considered those that are below 100 nanometers. In this case, we will be addressing mesoporous silica dioxide nanoparticles. These nanoparticles are pretty special for a number of features. Firstly, they can be tuned down, meaning we can synthesize them from 10 nanometers to more than 60,000 nanometers. It really depends on the process of the synthesis method. These nanoparticles are also very special due to their high surface area, their thermal stability, and inherent biocompatibility, meaning they will not cause any adverse reaction with animal cells or human cells, which make them a suitable candidate to be nanocarriers for maybe drugs, a suitable candidate to remove maybe some transition metals. Will be addressing that. And there are suitable candidates also in environmental remediation. Okay, so firstly, the methods we use is the classical Stover method. In this case, we use a modified Stover method by introducing a surfactant, CTAF. In this Stover method, we need a precursor. In this case is the TEOS, the tetraethyl orthosilicate, which undergoes an hydrolysis. It is in water, dissolved in water, with a short alkoxylane. Maybe in this case we use we could use isopropanol, ethanol. Okay. Furthermore, the TEOS, after the hydrolysis, it condensates in silicon tetrahydroxide, and there is when the nucleation and growth begins to happen. This nucleation and growth happens with the CTAF, and after 2 hours, this is really standardized for obtaining nanoparticles of around 100 nanometers. In this case, we obtain nanoparticles of roughly 95 nanometers. We for every nanoparticle or nanomaterial synthesis, we always obtain a size distribution. There are not always the same size. Okay, so for the results, we obtain these spherical nanoparticles. In this image, in figure 1, image A, we can see the nanoparticles with a rock surface. The same microscope, the scanning electron microscope, does not have the resolution to see the pores. The pores made by the CTAF, by the surfactant. This is due to the electrostatical interactions between the surfactant and the silanol groups in the surface of the nanoparticle. So we were asking, how can we observe the pores, the mesopores? In this case, the UPAC defines mesopores as those that are between 2 and 50 nanometers. So we use a transmission electron microscopy. In this case, this is the image B, and we can see very clearly the pores. But we were not sure that the pores had the size and the distribution we needed. So we made another experiment with the BJH and the BET methods. Those are methods that use nitrogen adsorption in the surface of the nanoparticles, and we can get to know the size of the pores, the distribution of the pores, and also the surface area. We obtained this um, adsorption isotherm from the nanomaterial. It is characteristic of a type 4 material, which means it's a mesoporous material. That was exactly what we were looking for. And finally, for this page, from this section of the poster, in figure 2, image D, we can observe the same nanoparticles, but these underwent another process. We charge them with iron in order to test if this transition metal can not also or can or cannot interact with the surface of the nanoparticles, but also occupy these pores. This is really, really important to us because we pretend to utilize these nanomaterials in an in vivo application. And in this case, we can see a clear difference in image D with the iron. We cannot see all the pores in all the nanoparticles, which could be an indicator of iron occupying these pores. So in order to address that, we also made an EDS, it is an experiment that can be made with the SEM microscope. So in this EDS, we were looking for the element analysis, the elements that compose these nanoparticles. It's mostly silica and oxygen, as we expected. We have a little bit of iron, of aluminum, sorry. This aluminum was used as a substrate for the nanoparticles in order to get them into the microscope. And a little bit of iron. Well, we were expecting more iron here, but we have to consider that iron is only in the surface and not in all the nanoparticles. So we asked ourselves, how can we address this iron adsorption? So we use an organic material that is known as phenanthrolene. This phenanthrolene forms organometallic complex with transition metals. In this case, we had to wash everything very well with distilled water, with molecule water, in order to avoid any traces of other transition metals. And we used the purified iron. For this experiment, we left the iron and the phenanthrolene react, and we left them for 45 minutes in a standard spectroscope, and we measured the absorbance during that time. So the diminution of absorbance is an indicator that the whole complex is entering the nanoparticle. But we were not convinced by those results, so we used another, maybe it is a not so orthodox method for this kind of nanomaterials, but we use AFM, that is atomic force microscope. This microscope has an atomic resolution, as the name implies. And for this case, we have two materials again, the one with iron and the one without iron. The one with iron exhibit a smoother surface with not so much of the pores visible as we saw in these images of the TEM images. And the nanoparticles without iron exhibit a higher roughness. This is a significant difference, which is an indicator that iron is indeed occupying these pores. So we are trying to really address every doubt that we had within nanomaterials in order to use them in biological applications. So addressing that, it is crucial for us that in vitro applications, everything that is used, everything should be sterilized, and these nanoparticles definitely can be sterilized by autoclave. Is the common method to do this. And after sterilizing them for 21 minutes, in the autoclave, 120 Celsius, and we left them in cell culture media, is the F12, It is the standard for many cell cultures, from specifically for cellular lines. Yes. We left them for 2 hours at conditions, 7 Celsius, CO2, and there were absolutely no microbial growth, no bacteria or fungi, anything, no pH changes. This is very relevant because it means we can, and we are currently using these nanoparticles for in vitro applications. But in this poster for application, we will only be addressing the characterization of nanoparticles. Where can we use these nanoparticles? Can be applications, it can be for nanoparticles, for drugs. We also have seen them in other articles, in other references, where they use them with the idea of cleaning water from dyes like clean blue. We already talked about the implications of this. We are looking forward to see you at the Congress, and best regards.